Hi everyone, I'm Dr. Babita and in this video we will talk about the layers of retina. It is a complicated topic and I've tried my best to explain the anatomy of retina in the simplest way possible. I've also posted the Hindi version of this video on my other channel, the link to which you will find in the description box below. So let's begin. To understand the layers of retina and their function, it is important to first understand the basic structure of the eye. The eye, as you know, has three coats, an outer coat, a middle coat, and an inner coat. The outer coat is composed of the cornea in the anterior 1/6 part, while the posterior 5/6 part is made up of sclera. Internal to the outer layer lies the uvea, which is composed of three parts from anterior to posterior. These are the iris, ciliary body, and choroid. And internal to the uveal tissue lies the retina, which is the innermost coat of the eye. So if we look at the posterior part of the eye, then the layers from inside to outside are retina, choroid, and sclera. This is the optic nerve, and this space in front of the retina, which is filled with vitreous humor, is known as the vitreous cavity. Light enters the eye through the pupil, and if the path of the light is clear, then it eventually falls on the retina. Retina processes this light signal into electric signal through a process called the phototransduction cascade. Then this electrical signal is carried to the brain via the optic nerve. And finally, the brain processes this stimulus into an image, and this is how we are able to see. This is the structure of the retina. Retina has 10 layers in total. The outermost layer that is in contact with the choroid is known as the retinal pigment epithelium or RPE. Then comes the layer of photoreceptors. We have two types of photoreceptors, rods and cones. That is why this layer is also known as the layer of rods and cones. Then comes the external limiting membrane. Internal to that is the outer nuclear layer, then outer plexiform layer, followed by the inner nuclear layer. After that comes the inner plexiform layer. Eighth layer of the retina is the ganglion cell layer. Then is the nerve fiber layer, and the innermost layer, which is in contact with the vitreous, that is the 10th layer of retina, is the internal limiting membrane. Let's discuss these layers in detail. The outermost layer is the retinal pigment epithelium or the RPE. Outside of it lies the choroid, and internal to it is the photoreceptor layer. Retinal pigment epithelium is a monolayer, which means that it is a single layer of hexagonal shaped cells. The RPE cells are cuboidal in cross section, and two adjacent RPE cells are joined by tight junctions called zonullae occludens. It is important to know this because these tight junctions form what is known as the outer blood retinal barrier, because they make the retinal pigment epithelium impermeable to water and ions by blocking the extracellular movement of water and ions and other macromolecules between the choroid and the retina. We will talk more about RPE later, but before that we need to first understand the layer that lies internal to it, which is the photoreceptor layer. When light rays fall on the eye, it passes through the cornea, aqueous humor, lens, vitreous cavity, and through the inner layers of retina to reach the photoreceptor layer. Light is composed of photons, and photoreceptors are the specialized cells of retina that are capable of detecting and perceiving these photons, and then they convert them into signals, and this eventually helps in vision. That is why these cells are known as photoreceptors, because these are the cells of retina that can detect and respond to light. Through a complex process called the phototransduction cascade, this visual signal travels through the various layers of retina and eventually reaches the brain through the optic nerve. As we all know, our eyes have two types of photoreceptors, rods and cones. Rods are responsible for dim light vision, while cones function in bright light and are also responsible for color vision. So the structure of a photoreceptor can be divided into 5 parts: an outer segment, an inner segment, a cilium, an outer fiber and an inner fiber. This is the outer segment. The plasma membrane of the outer segment is arranged into hundreds of flattened discs. As you can see these discs here, the names of these cells are derived from the shapes of their outer segments. So this outer segment looks like a rod, so it is known as a rod cell, while this outer segment looks like a cone, so it is known as a cone cell. The outer segments of cones are shorter and wider than the outer segments of rods. These flat discs of the outer segments of photoreceptors contain photopigments. The photopigment present in rods is known as rhodopsin. Rhodopsin absorbs light maximally at 500 nanometers of wavelength. Humans have only one type of rods. The photopigment present in cones is known as the cone opsin. There are 3 different types of cone opsins. Therefore, we have 3 different types of cones: L cones, M cones and S cones. L cones absorb light maximally in the red part of the visual spectrum. M cones absorb light in the green part and S cones absorb light in the blue part of the spectrum. The wavelength at which light is absorbed by each type of cone is mentioned next to it. So the layer of rods and cones lies internal to the retinal pigment epithelium, which is the outermost layer of retina. The outer segments of these photoreceptors are located between the microvilli of the retinal pigment epithelium, and this is known as the interdigitation zone. These small projections of RPE cells that you can see here are the microvilli. There are no junctional complexes between these RPE cells and the photoreceptor outer segments, and therefore in various pathological conditions, these two layers can get separated to give rise to a subretinal space where fluid or blood can collect. These outer segment discs are produced at the base of the outer segment. This is the base here, and as new discs are formed, they move towards the tip of the outer segment. Each disc is then shed and phagocytosed by the RPE cells. Next to the outer segment is the inner segment. The inner segment of a photoreceptor is divided into 2 parts. The outer zone, it is packed with mitochondria and is known as the ellipsoid zone, while the inner zone contains smooth and rough endoplasmic reticulum and it is known as the myoid zone. Cilium, as you can see, is this connecting stalk that joins the outer segment with the inner segment. So now that we have learned quite a bit about the photoreceptors, it is a good time to discuss about the functions of the retinal pigment epithelium. RPE cells are essential for photoreceptor health. They are involved in the phagocytosis and degradation of the shed outer segment tips, as we just learned. They also play a role in vitamin A metabolism and in its regeneration in the visual cycle. They contain melanin granules which help in light absorption and prevent scattering of light, and thus they prevent the retinal layers from light-induced damage. They secrete the matrix that surrounds the photoreceptor inner and outer segments. They are also responsible for the active transport of materials between the choriocapillaris and the subretinal space. Photoreceptor density varies across the retina, but on an average, each RPE cell lies in contact with 45 photoreceptors. Coming back to the structure of a photoreceptor, the nucleus of the photoreceptor lies inside the cell body. Then there is an outer fiber and an inner fiber. The outer fiber connects the inner segment to the cell body, while the inner fiber is an axonal process. It arises from the cell body and ends in synaptic terminals, which contain synaptic vesicles. In the case of rods, this synaptic terminal is called a rod spherule, while the synaptic terminal of a cone is called a cone pedicle. Here it is very important to note that when we talk about the second layer of retina, which is the photoreceptor layer, we are only talking about the photoreceptor outer segments and inner segments. The rest of the photoreceptor structure is not included in this layer, which will become clearer to you as we will move forward in this video. Moving on, the third layer of retina is the external limiting membrane. The external limiting membrane is not a true membrane. It is made up of intermediate junctions between the rod and cone inner segments and the processes of Mueller cells. These skin-colored cells that I have made here are the Mueller cells. We'll talk about what Mueller cells are a little bit later in this video, but as of now, this information should be enough. So this is the external limiting membrane. Integrity of this membrane is important in the protection of photoreceptors and in maintaining visual acuity. So the inner segments of rods and cones pass through this external limiting membrane to reach their cell bodies. These cell bodies of the photoreceptors are present in the next layer, which is the outer nuclear layer. So the fourth layer of retina is the outer nuclear layer, which is formed by the cell bodies of photoreceptors, and as we just learned, these cell bodies contain the nuclei of these photoreceptors. So outer nuclear layer is nothing but the nuclei of rods and cones. The cell body of each rod and cone has two processes that travel radially in two different directions. One of these processes reaches the inner segment as we just discussed in the previous section. The other process is the axon, which travels vertically to reach the next layer, which is the outer plexiform layer. So the fifth layer of retina is the outer plexiform layer. The outer plexiform layer contains the synapses of the rod and cone photoreceptors with the dendrites of the cells that are present in the layer above it. Those cells are the bipolar cells and a few horizontal cells, which are present in the inner nuclear layer. Thus, outer plexiform layer is the first step in the processing of light signals because this is where the first synapse is taking place. So because of the presence of these synapses, this layer conducts signals from the outer nuclear layer to the inner nuclear layer. So above the outer plexiform layer lies the inner nuclear layer, which is the sixth layer of retina. So just like the outer nuclear layer, this layer also contains the cell bodies and nuclei of certain cells. The cell bodies of 5 different cell types are found here: bipolar cells, horizontal cells, interplexiform cells, Mueller cells, and amacrine cells. The cells nearest to the outer plexiform layer are the horizontal cells. The amacrine cells are present near the inner plexiform layer, which lies above it, while all the other cells are located in between. Bipolar cells are the cells we are mainly concerned with. Bipolar cells receive input from the photoreceptors through synapses in the outer plexiform layer. Then they process the light signal, and the axons of these bipolar cells terminate as synapses onto the cells that are present in the layer above them, which is the ganglion cell layer. Thus, bipolar cells are the first order neurons in the phototransduction cascade because they are the first to synapse with the photoreceptors. There are 13 types of bipolar cells which can be subdivided into two categories. So there are rod bipolar cells which synapse with rods, and there are cone bipolar cells which synapse with the axon terminals of cones. Other type of cells present in the inner nuclear layer are the horizontal cells. They form the outermost row in the inner nuclear layer, and just like bipolar cells, their dendrites also synapse with the photoreceptor axon terminals. There are also amacrine cells which form the innermost row in the inner nuclear layer. The other cell types found in the inner nuclear layer are the interplexiform cells and the Mueller cells. These are the Mueller cells. We made a mention of Mueller cells when we were discussing about the external limiting membrane of retina, and we will talk about them a little more in detail a bit later in this video. The next layer is the inner plexiform layer. This layer contains synapses between the cells of inner nuclear layer with the cells present in the layer above it, which are the ganglion cells. So this layer mainly contains synapses between the bipolar cells and the ganglion cells. So the next layer of retina is the ganglion cell layer. This layer contains the cell bodies of retinal ganglion cells or RGCs. Ganglion cells are the second order neurons in phototransduction cascade because, as we just learned, they synapse with bipolar cells in the inner plexiform layer. RGC is the main output neuron of retina because through them the visual signal travels out of the retina to further travel through the optic nerve, We will see how that happens. So there are approximately 0.7 to 1.5 million ganglion cells per retina in a young adult. Besides ganglion cells, a few amacrine cells are also present in this layer, which are known as displaced amacrine cells because these amacrine cells are displaced from their original location, which is the inner nuclear layer. There are approximately 20 different RGCs, and it is interesting to note that about 1 to 2% of these cells are intrinsically photosensitive, which means that just like the photoreceptors, rods and cones, these intrinsically photosensitive ganglion cells can also perceive light. Each ganglion cell has a single axon. Each axon makes a 90-degree turn like this to reach the inner surface of the retina. These axons are unmyelinated so that they cannot interfere with incoming light, allowing the light rays to reach the photoreceptor layer. So these axons of ganglion cells make the 9th layer of retina, which is the retinal nerve fiber layer or RNFL. Ganglion cell axons travel from all parts of the retina towards the optic disc to form the optic nerve. Axons from nasally located ganglion cells project directly to the nasal side of the optic disc to form radiating fibers. Axons from ganglion cells temporal to the fovea form arcuate fibers that course above and below the macula and reach the optic disc at its upper and lower edges. Axons from ganglion cells between the disc and macula course directly to the disc and form the papillomacular bundle. The 10th and the innermost layer of retina is the internal limiting membrane. This layer covers the inner surface of the retina. It is made up of the terminal ends of Mueller cells and astrocytes. We will talk about these cells in the last section. So these are Mueller cells, and this is how they form the internal limiting membrane. Being the innermost layer of retina, the internal limiting membrane is in contact with the vitreous. So these were the 10 layers of retina, but apart from these 10 layers, retina also has some supportive cells which are known as glial cells. Retinal glial cells fill the extracellular space and provide physical support to the retina. These cells also maintain the ionic environment around retinal cells. They play an important role in glycogen storage, in the reuptake of released neurotransmitters, in the removal of debris, and in insulating axons and neurons. There are 3 types of retinal glial cells: Mueller cells, which are radial glial cells, astrocytes, and microglia. The most abundant glial cells are Mueller cells. Mueller cell bodies and nuclei are present in the inner nuclear layer along with bipolar cells, horizontal cells, amacrine cells, and the interplexiform cells. These cells are spread across the layers of retina from the innermost layer to the photoreceptor layer to form the cytoarchitectural support of retina. At the level of the photoreceptors, the outer processes of Mueller cells form the external limiting membrane, while the inner processes extend to the inner surface of the retina, forming the internal limiting membrane. So basically, these Mueller cells virtually fill in most of the space of the retina which is not occupied by the neurons. Other glial cells are astrocytes, which are star-shaped fibrous cells. They are usually present in the nerve fiber and the ganglion cell layer. Retinal astrocytes have processes which insulate retinal vessels and axons. And the third type of glial cells are microglia, which are tissue macrophages. The microglia become activated in damaged or degenerating retinas, where they function as scavengers of debris and may also function to present foreign antigens to T lymphocytes. So with that, we come to the end of this video. Let us revise the layers of retina once again. There are 10 layers in total. From outside to inside, first comes the retinal pigment epithelium, then the layer of photoreceptors, then external limiting membrane. Internal to that is the outer nuclear layer, outer plexiform layer, followed by the inner nuclear layer, inner plexiform layer, ganglion cell layer, then is the nerve fiber layer, and the innermost layer that is in contact with the vitreous is the internal limiting membrane. Photoreceptors are sensory receptors, bipolar cells are first order neurons, and ganglion cells are second order neurons. There are 5 main layers: the outer nuclear layer, which contains the cell bodies of photoreceptors, the inner nuclear layer, which contains the cell bodies of various cells, but the most important cells are bipolar cells. And then there is the ganglion cell layer, which mainly contains the retinal ganglion cells. The other two important layers are the plexiform layers, where synapses between these nuclei occur. Outer plexiform layer contains synapses between the cells of outer nuclear layer and the inner nuclear layer, while the inner plexiform layer contains synapses between the cells of inner nuclear layer and the ganglion cell layer. Besides these, there are three types of glial cells, which are the supportive cells of retina, the most important of which are Mueller cells, which form the internal limiting membrane and the external limiting membrane. Do like and share this video with your friends and colleagues if you found it useful, and do subscribe to my channel to support free education. Thank you very much.